Hi, Roy Treveline here, AP Bio, Next Sacred Lab. We're looking at genetics. And we're going to be doing a little kitchen biology because we are not in lab. Anyway, we're going to look at the genetics of corn. Corn's a big deal. Its scientific name is Zea mays. Zea mays is a big deal because here in the Midwest, that's what we grow and export. We grow a lot of corn. And we're going to look at two different traits today. The first traits we're going to look at are the difference in corn color, the kernels. Each one of those corn kernels we're going to look at in terms of a fruit. And we want to know the phenotype or the physical characteristic of those. So what I'm going to send you home with is a bag of corn kernels. And I need you to realize that what we're going to cross is the F1 generation. So if we look over here at this card, we see that the first generation, these ones that I bought, and I need these back, by the way, because it gets a little expensive to buy them. Right? We crossed purple and yellow, and the F1 generation is all going to be purple. Now, it's okay to do with corn plants. We're going to cross brother and sister, but do not try that at home. We're going to cross brother and sister, and we're going to get what they call an F2 generation. This is a monohybrid cross, a single trait, and we're going to cross two heterozygous individuals. When you draw these Punnett squares, make sure you exaggerate if you're going to use P's so that you can see the difference between a capital and a recessive allele. And when we cross these two for a monohybrid cross, we're going to get one homozygous purple, two heterozygous purple, and one yellow kernel corn. In other words, a 3-to-1 ratio, very similar to what Mendel ended up with his pea plants for genetics. And the hypothesis would go something like this as we practice writing deductive reasoning hypothesis. A hypothesis would say that if the dominant allele for purple if the allele for purple, is dominant over the yellow allele, and we cross two heterozygous individuals, then the F2 generation should have a 3-to-1 purple to yellow ratio, a phenotypic ratio of a 3-to-1. And if we looked at all these corn kernels I'm going to send you home with, your job is going to be to measure those out, right? So I just took all my purples and put them on one side. I put all my yellows on the other, right? Purple, yellow, and so on and so forth. And what do you expect? If I sent you home with 100 corn kernels, what do you expect? How many purple to yellow? Well, these purple ones have a genotype of either big P, or big P, little p. And the yellow ones, remember, are only going to be little p. They're going to be homozygous recessive. Well, if I sent you home with 100 kernels, we would expect 75 to be purple, 25 to be yellow. 75 purple, 25 yellow. And then what I'm going to ask you to do is to use a little bit of statistics, something we did with the M&M lab at the beginning of the year, and calculate the chi-square, right? How close are we from the observed to the expected? Observed minus expected squared divided by expected. Then we're going to do class data. I'm going to get all our data together. We'll put it up on a spreadsheet. And we should see the class data being a little more statistically significant than our individual data. Again, I'll send you home with the corn kernels, or if you can come pick them up. If for some reason you cannot, then the rest of us will pick up the slack and count more corn kernels. But it'd be much easier if all of us just did some. Okay, I'll send you home with a small bag. Now, the second thing I'm going to ask you to do is to make a dihybrid cross. In case of a dihybrid cross, we're looking at two traits. Again, these individuals would have started off purebred. In other words, we'd have crossed purple with these smooth, so it'd be big P, big S, and we crossed it with a little p, little s, and we'd get all heterozygous individuals in our F1 generation. We take those siblings, these heterozygous individuals in the F1, and my question to you is, what would we get in the F2? What would our F2 generation look like? So I brought a corn kernel in here that I'm going to send you home with. I think probably the easiest thing to do, if you guys come up with a better way to do this, let me know. But I think what the easiest thing is going to be is to actually just measure or just put a little mark on each of the corn kernels as you go, so you know which ones you counted and which ones you didn't count. But if you look, there's going to be several phenotypes here that you need to count, actually 4, right? There's going to be some that are totally yellow and smooth, and some that are going to be yellow and wrinkled, some that will be smooth purple, and some will that be wrinkled purple. So there's 4 phenotypes in this dihybrid cross. Now, we know that the big P stands for purple, little p for yellow. What does the S stand for? Well, the S is standing for the amount of starch in the content of those corn kernels. If we're looking at a corn kernel with lots of starch, it's going to stay nice and smooth. Starch, if you may recall, is a polysaccharide, big sugar, right? But some of the corn kernels have a recessive allele that contains a lot more simple sugars than polysaccharides. A simple sugar is going to taste sweeter, and what it's going to look like under your vision is it's going to be these wrinkled seeds here. So you're going to take your marker, you're just going to come along and say, oh, there's smooth yellow. If you had to help with somebody, that'd be awesome. Here's wrinkled yellow, right? So this one's going to be little p, big S, little s. This one's going to be little p, little s, homozygous recessive for both traits. Now I'm going to try, and again, you just whiteboard marker or some type of marker, you can just kind of count them as you go. So hopefully somebody come up with a better way, let me know. But hopefully you'll come up with a way to count your one corn kernel. And again, we're going to do the individual data versus the class data. And I will ask you to do chi-square. So, here's an individual cross. Again, we got the heterozygous for the P's, heterozygous for smooth and wrinkled, just like Mendel did. A little close up. So here's the 4 phenotypes you're going to look at. Here's a round, smooth purple, right? Here is a purple wrinkled, right? More sugar content, higher sugar content. Here we see a smooth yellow, and here we see a wrinkled yellow. Those would be the 4 phenotypes. And the relationship we're going to look for is called a 9-to-3 to 3-to-1. Now again, when we make our Punnett squares, people always assume they know how to make them. What determines the size of your Punnett square is the number of gametes, the number of sex cells. If these individuals are heterozygous for both traits, then there are 4 possible sex cells for dad: big P, big S, big P, little s, big S, and little p, little s. And the same is true for mom, 4 possible 4 possible gametes. And here are our possible children. 16 offspring are ending up in this Punnett square because it's a 4x4 Punnett square. Now, these light blue ones are representing individuals with all dominant traits, dominant phenotypes. They would be purple and they would be smooth. 3 out of 16 are going to be these red guys, where they're going to be purple, but they're going to have the recessive allele for the amount of sugar content. They're going to be a little bit sweeter. We see 3 over here in green that are going to be yellow because they're little p's, but they're going to have at least 1 big S, and so they're going to be starchy, which under your, when you look at them with your eyes, you're going to see a smooth appearance. And then 1 lonely guy here that's going to be homozygous recessive for the P's, as well as the, homozygous for the color, as well as homozygous for the sugar level. And again, we're going to crunch our numbers. We're looking at observed minus expected. The basic gist of this, just like we did with the M&Ms, is we want to know how close we are from the observed to the expected. So if we go back to my simple equation with the 3-to-1 ratio, and we had 100 corn kernels, we expect 75% to be purple, 25% to be white. Remember, this funky little number here is the sum of. So we will do observed minus expected, count your corn kernels, square that, divide by the expected, plus the other phenotype, so purple and yellow. And we'll get a number You look that up on the chi-square chart, and it will determine whether or not our data is significant. And I'll send you out some information on that. Okay, Sacred Lab Genetics. Thanks, and thanks for those of you that are picking up corn kernels. Bye.